All right, so here we go. So in your, in the bottom of week 6, I have some notes here. Some people like the written stuff. Some people just want to hear me talk. You can do either or. We're going to basically learn something about Hardy-Weinberg principal, equilibrium, and microevolution today, and do some calculations. And at times it appears very, very difficult when you first learn it. But hopefully through today's lesson, that everything could be crystal clear and say, wow, I think this is pretty easy now. Okay, so give me a shot at this, and you see. People thought that when you first heard about chi-squared, it's impossible, right? But when you stop and actually did it a few times and saw it different ways, you got pretty simple. So let's talk about a microevolution. So what is microevolution? Because we know evolution is essentially, and we saw this in cladograms, where we have a divergent evolution of new characteristics. You know, for instance, we had the amniotic egg, okay, and then maybe we had a different type of skull. And of course, as we got more complex, we added more characteristics. And that occurred because of a mutation. A mutation to our existing genome changed some proteins that allowed for these changes to occur. Or it changed a group of genes by changing a protein or a regulatory gene that changes others. We saw that with the teosinte and maize. We saw that with the stickleback. So sometimes these major changes could be a single nucleotide mutation, or it could be a single change in an amino acid, as we talk about this, that changes a single protein that changes a whole bunch of genes. It doesn't really matter, but we have a direct descent from simple to more complex. And as I said before, normally we keep these simpler adaptations because they're still very helpful to us. Okay, you can't be a complex animal unless you have the basic needs covered. So it's pretty logical. Now, as we move and descend from simple to complex through these major characteristics, that takes a long period of time. And that's the difference between microevolution and regular evolution. Now, we've seen evolution, and I've given you examples. For instance, I've given you the example of the beaks in the Galapagos Islands in the 70s. There's researchers who saw in a drought period the significant change of the beaks because the food changed. There was different selective pressure to allow the birds to survive with bigger beaks when the nuts were bigger, the food was bigger. The smaller beak birds couldn't do that, so they died out. But when there was a monsoon season, we saw there was a significant decreased size of the beaks, again, because of new selective pressures. So we've seen it happen pretty fast, but generally speaking, this descent is slow because it requires a mutation, and mutations occur very slowly. So a lot of times, we want to look at our population to see what the trends are. How are we changing? Now understand this, as a modern day population, humans, where we control our environment, so to speak, okay, we don't have the same selective pressures that we used to have. Okay, I'm not going to be selected against because I'm so tall and I stick out in a crowd. I mean, I'd probably be picked out easier by a predator, okay, being this height. But I'm not selected against for that. I'm not selected against because I'm nearsighted. I can't see I can't hunt as well. All those things we can correct for. Even if I have some disorder that runs through my family is able to survive because of modern medicine. So we don't have those kind of selective pressures. But we still want to know if there is a trend. Is there something changing? So what microevolution is to look at a simple single allele in a population. See, evolution, you're looking at all the major changes. We want to look at a single allele from a single gene to see if that frequency, and I want to make sure we understand this, the frequency is how often, okay, that allele is expressed in the population. So microevolution is about looking at a single allele. Okay, we've kind of done this already, but on a bigger scale. For instance, we looked at the normal distribution of our peppered moth. Okay, we knew down here that some on the outskirts, okay, were selected against originally before the Industrial Revolution. We had some individuals on this blue line in low quantities that had black wings, but they didn't get to survive because they were selected against. They were easy prey against a light-colored bark. But when there was a new pressure or change in the environment because of the soot of the factories and made the bark darker, okay, and then these guys were selected against and these were selected for, and we had a shift. And we were able to see in our research article for the test 1, there was a significant change. So when we talk about evolution from here to here in a cladogram or a phylogenetic tree, we're talking about after we what? Created the new normal distribution. But in microevolution, we're after, hey, when did this start to change? When was there a start? Okay? And there's a reason for that. If I'm going to study population, I don't have 1,000 years, and no one cares what's going to happen in 1,000 years. They want to know what's happening now. Case in point, if I have a certain disease that's recessive, there's plenty of recessive disorders. Why do you think there's plenty of recessive disorders? Mutations that do what to a certain working protein? It changes its shape so it can't do what it's normally supposed to do. So some diseases are diseases because the person's homozygous recessive for that allele, and a protein that was supposed to be made because they have both pairs from mother and father that is a broken gene, a recessive allele, all right? They can't make that protein. So whatever function that was important for that gene is not being taken care of, and they're expressing that. Hemophiliacs are like that. Hemophiliacs are essentially people that if they have a simple cut, they don't have a clotting factor. They don't have a simple protein to clot, so they will bleed to death. Okay, and that's a very famous line of evolution that we've actually studied because there was royal families that kind of just breeded with themselves, and we have a way of tracking that. It was kind of the earlier type of genetic research that we know about. So hemophiliacs are a homozygous recessive, so they have two. So the point being is that, boy, why don't I can track if there is more or less hemophiliacs in my what? In my big population. Because now you may say, why do I care about that? Well, I may care about that because I may be a hospital that has to have a certain amount of medication always at hand for people who come to my emergency room. If I know the percentage of people in the population that are hemophiliacs, then I better be ready for that certain percentage to come in and need that clotting factor or whatever that might be. But what if we're evolving? What does evolution mean? It means that we're making possibly a mutation that makes a new allele or a new disorder, right? But that takes a long time, right? Mutation. So generally speaking, mutations don't really happen in our generation. They happen once every multiple generations. So that's probably not going to happen from year to year. But what I will know is that evolution is a changing of the frequency of the allele. Are there more hemophiliacs next year? Something we need to know. Where is the trend? Are we going up, down, or is it staying the same? Now, if there's no evolution, we would expect it to what? Stay the same. Now the question is, how do we test for that? And that's where Hardy-Weinberg comes in. We need to have some kind of null, some place where we know where there should be no change. And then when we test for it, hey, is it the different or the same as what we have? So Hardy-Weinberg was a way of developing, if there is no evolution, we can actually calculate how many individuals in society, okay, are hemophiliacs, and we can predict the next generation and the next generation, which by the way will be what? The same as our current generation, because there's what? No evolution. Okay, so stay with me with that. So, we're dealing with Mendelian genetics, okay, and we know that it was argued in the 1900s. And the major problem of Mendelian genetics, of course, some people argued, and again, we know that Gregory Mendel earlier developed Mendelian genetics. It was brought back in the early 1900s, and one of the problems people had, and before the early 1900s, of course, there was no DNA. People were still trying to figure out what the hereditary factor was. They knew there was something to hereditary factors, so they didn't know exactly a lot about genetics. But they did know that, hey, my son and daughter have similar traits as I do, and there is something to the fact of this hereditary. And so knowing Mendelian genetics, people argued that Mendelian genetics don't work because if I've got a dominant trait, shouldn't that always dominate our genetics? Because if you think dominant recessive, now we know recessive means a broken gene. We didn't know that then. So if you have a recessive gene that's what? Weaker to the dominant, from generation to generation to generation, you'd expect the dominant allele to what? To grow, even without evolution, because it's the what? The dominant one. Now we know what that means. They didn't know what that meant. We now know because I made you learn this through what? Chromosomal line of inheritance. Dominant just means you're working, and the recessive means you're not, correct? But they didn't know this. So if you're going to call it dominant, hey, if I've got the red and a white allele, the red's always dominant for the flower. Hey, that's the stronger what? Hereditary factor. Shouldn't that dominate over time, and over time we're all going to have red flowers? That's what they thought. So people argued that, and that kind of messed up. Now, there was a guy named J.H. Hardy. He was a total mathematician. He was actually played cricket with some biologists who had this problem of, hey, dominant alleles should be dominating. And he just sat down, and suppose he did it in 15 minutes, he figured out that, no, it should stay the same. And he scrabbled it out, and he says, I don't know anything about biology, but just like, listen, mathematically, it stays the same, and here it is. And he didn't think nothing of it until everyone's like, oh my God, it was the best thing since sliced bread. Unless you know about sliced bread. You guys know about sliced bread? Okay. So Hardy was a mathematician who figured out what the allele frequencies would be from year to year. I'm going to show you that, okay, if there was no evolution. Okay, and again, we're in a society where we're not really evolving, but in truth, there could be frequencies of alleles that are changing, and that by itself is called what? Evolution itself, microevolution. And we want to know, because if the hemophiliacs, I'm just giving you an example, are increasing, then we need to make sure that our hospitals, emergency room, have that amount, whatever that might be, calculated for our population in the area to be available there. Okay? That's part of modern medicine. And that's just one example of why we'd want to know. Okay? So he said that I found a way to find what that null is. Oh my gosh, null hypothesis? No, but what the no change values would be from year to year if there's no other forces at play. Okay, and I'm going to show you that. His work was brilliant. He said it was the least thing he's ever done in his life, but I don't know why it's so big deal, but they gave him the name Hardy. Now someone else, Weinberg, was a German guy who independently in the same year came up with the same math. And the math is simple. I'm going to show you how they did it, but I guess it was revolutionary at the time. Okay, now, the point is that they call it Hardy-Weinberg Equilibrium because equilibrium by the word means nothing happens. Now we're in chemistry, and if I have a soda bottle, let's just say it's a water bottle. And why it looks like this is because I wanted it off-centered because I wanted to be artsy right there. Now, if I have some water and I have a stopper, we know when we look at a water bottle with the top on, the line of water is never going to change. Okay, probably not going to have a wave right there, right? It's going to be straight across. Now, if I would take my eyeball and look at it, it's not going to change. And the reason why is, as fast as some of this water becomes a gas, evaporation, some of this condenses back to a liquid. So the rate of evaporation, liquid going to a gas, is as fast as the what? Gas going back to a liquid. That has to be true. It's happening. But because that rate is occurring, then we're at the same amount of water. Take the top off, the rate of what? Evaporation is faster. Now there's some condensation, but because the gases are leaving, can they condense back into the liquid? No, and so what happens is this what? Goes down. And you guys know that. So equilibrium means that the values are staying constant because there's equal changes. So Hardy-Weinberg equilibrium, if we're not at what? A point where we're evolving. Right? There's no selective pressures, right? No other forces at play. The people who are dying of a certain allele have to be equal to the ones that are being born with that allele to replace them. Right? So we're replacing the alleles that are lost through death, through the birth. Okay, now, see here are the forces that are at play, okay, that he talked about, that in order for his no change, no evolution, no change of frequency to be true, we have to discuss these. Okay? So to be at Hardy-Weinberg equilibrium, that no change of just even one allele frequency. Frequency, by the way, means what? You have to know this. Frequency is the probability that that allele is expressed. What if that allele is always expressed? What's the probability? 100%, but we're not going to use percentage, use 1. What if that allele is expressed half the time? Point. Good. So make sure we understand frequency is going to be some number less than 1. Okay, what is the probability of flipping a heads? 0.5. Heads and tails both? 1. Okay, so we're still using our probability laws. Okay, now, what are these forces? Okay, if this is happening, we're going to have changes of allele frequencies, which means we're going to have evolution. microevolution. First of all, small populations. If we have a small population, it's a bad thing. Chance events. If I flip a coin 1,000 times, I could probably predict that about what? Pretty close to 500 would be tails, right? Heads. The more that I flip it, the less chance events affected. If I flip it 10 times, isn't there a good chance I can have 7 out of 10 or 8 out of 10? How many families do you know have all girls, all boys, when there's a 50-50 chance? So the smaller the population, the more random events can mess with allele frequencies. If someone dies, if you have 10 people in your population, and just by chance, everybody who's homozygous recessive died, what's that do to the frequency of the recessive allele in your, it goes down. If you've got a huge population, that prevents it. Of course, we have a huge population, so small populations will mess this up. What else messes up? Selective mating. Now, we don't have this in our population. We don't say, hey, the only people who get to have families are the tall guys. Okay? Doesn't happen. Everybody mates. Everybody has kids who wants them. We don't have selective mating. You may say, well, I select him because he's so cute. Okay? But we don't, but everyone's level of cuteness or whatever might be like, oh, well, the point is, we don't just say certain parts of society breed and others don't. And in real life, animals do what? Prefer who they breed to based upon some ritual, okay, maybe some fight with the horns. We don't have that. We don't have selective mating. We select who we love and have families with, but everybody has equal chance on everybody. So selective mating would cause evolution because if we only liked blonde-haired men, then we would lose the other alleles, and we don't do that. Okay, what else? Mutations would mess this up, but we know mutations are slow. Slow. For as long as we live, how many generations we need, this is not going to mess things up. How about gene flow? In and out. They call this gene flow, in and out of a population. If I have a small population, and then what? Individuals come into my population with different alleles, won't that mess up my allele frequency? Absolutely. That would be evolution. Okay? And of course, no natural selection. There can't be a selective pressure for an allele to happen. And again, because we live in a modern day society, there is no selective pressure. So these are the things that would mess up Hardy-Weinberg. So anytime we do Hardy-Weinberg Equilibrium to figure out what the allele frequencies will be if there is no mutation or small population or random mating, know what? Evolution, the allele frequencies can be predicted from your population to the next generation, or from a small random sample of population and apply it to the other, what? To the total population and its what? Next generation. Okay? So, I said what would mess it up. So here are the criteria in order to use Hardy-Weinberg to figure out exactly what the alleles are if there's no evolution. You must have a large population so that there's a few deaths here and there, even unfortunately tragic, they don't mess up significantly any of the allele frequencies. No selective mating. You're not choosing one allele based upon he's tall or good looking or something. No mutations. There's no new alleles happening. Not a large enough influx of new alleles to the area, and of course no selective pressure. So this is important to realize. Any one of these 5 are occurring, you're going to have evolution. You're going to have the allele frequency change. Remember, evolution, yes, is new what? Major characteristics, but it's also just changing the current what? Frequency, probability of a certain allele. Okay, we were talking about having new alleles in our cladograms, but now I'm just talking about, hey, we're not developing a tail, we're not developing a third ear. So, but we're evolving microscopically, meaning there are changes in our populations of different alleles. And we want to know what they are. Okay? How do we know they're changing? We know what it is if it's not. Okay? So the Hardy-Weinberg kind of gives me that baseline. All right?